afternoon, dear students. This is Professor Sevmiş from Molecular Biology and Genetics Department. We are together with Physiology course today. Our subject is Digestion and Absorption of Food. Okay, let's begin with the definition of digestion. Digestion is the mechanical and the chemical breakdown of food into forms that cell membranes can absorb. And digestive system is consists of gastrointestinal tract, which is also known as alimentary canal, which is extending from mouth to the anus, containing mouth, pharynx, esophagus, stomach, small intestine, and large intestine. And also, in addition to gastrointestinal tract, the accessory organs which empty into the alimentary canal. So these are the tissues and organs, salivary glands, liver, gallbladder, and exocrine pancreas. Okay, so what are the functions of digestive system? Digestive system, basically the function is ingestion of foods and also digestion of large molecules into their absorbable molecular forms. And also absorption, nutrients from the gut into the internal environment, metabolic transformation of fuel molecules, and detoxification of foreign substances. And liver is very important for detoxification and conversion of the molecules into their untoxic forms. And also the digestive system is involved in immune functions. The digestive system fight against the infectious microorganisms which are not destroyed by the stomach acid. Okay, so what are the organs of the digestive system? The organs are okay, so the digestive system is very complicated system, and it begins with the mouth, and the digestion begins at the mouth, where the teeth break the foods into smaller particles, and then salivary glands, they secrete the saliva to start the chemical breakdown and also to moisten the food. And the contractions of the muscles in the face, they push the food into the pharynx. And then in the pharynx, okay, in the okay, so in the mouth, the muscles, they push the food into the pharynx, and then the soft palate blocks the upper pharynx not to enter the food into the nasal cavity, and also food passes over the epiglottis. Okay, so this is the epiglottis, which is the gateway between the digestive system and respiratory system, and food cannot enter into the respiratory system. And then food, now it is called as bolus, moves through the esophagus. So esophagus is the place where the pharynx and the stomach is connected. And then bolus reaches to the stomach. And when it reaches to the stomach, it causes a distension in the stomach wall, and causing the secretions of the hydrochloric acid and also the hormones which are involved in the digestion. And also by the aid of the stomach muscle contractions, so this food is converted into a thick liquid, so called as chyme, and then chyme enters into the small intestine through the pyloric sphincter. And the small intestine is the place where the most of the absorption occurs. And then the liver and the pancreas, they secrete their secretions into the small intestine to help the digestion. And the digested food is absorbed, and the remaining food enters into the large intestine. So the large intestine is the place where the digestion is complete and no digestion anymore, but the water and the mineral salts are absorbed by the large intestine. And then the chyme and the remaining chyme in the large intestine is converted into feces, and then this feces is stored in the rectum until the neuronal the stimulus comes, and then it is excreted with defecation. Okay, so of course the gastrointestinal system is regulated. So basically the regulation is done with regulated by the volume and the composition of luminal contents. So that means not nutritional state of the body. So this means that the body is designed to absorb all the nutrients that are ingested, whether or not the body really needs them to function. And so according to the luminal contents, the gastrointestinal reflexes are initiated by the luminal stimuli, such as distension of the gastrointestinal tract and chyme osmolarity. The chyme osmolarity is the total solute concentration in the lumen, and chyme acidity, also the chyme concentrations of specific digestion products like the carbohydrates, the fatty acids, peptides, amino acids, etc. Okay, so for the regulation of the gastrointestinal system, in addition to volume and composition, of course, there are two main regulatory systems. These are the nervous system and the hormonal system. The nervous system, the gastrointestinal system has its own neuronal system, which is called as enteric nervous system. So these neurons, the enteric system of neurons, have very short plexus. Plexus means the short branches. So these short branches make with interactions with each other, and then they have direct controlled regulation on the gastrointestinal tract. And in addition to this enteric nervous system, of course, the central nervous system has a regulatory system on enteric nervous system, and also direct regulatory system on the epithelial cells of the gastrointestinal tract. Okay, how about the hormonal regulation? In case of hormonal regulation, there are the secretory cells. So these are called as enteroendocrine cells. You see here, so this is a segment from the small intestine, and small intestine is covered with different various cells. So you see here in intestinal gland, and we see here the enteroendocrine cells. See, this is the enteroendocrine cell. The stimulus comes from the lumen, and then the secretions are released into the interstitial fluid, and from the interstitial fluid, the capillaries they absorb these hormones, and then hormones, they enter to the circulatory system, and then they can affect the other parts on the gastrointestinal system. Okay, so in the hormonal regulation, there are very important basic hormones which are secreted by the stomach and the small intestine. The main hormone secreted by the stomach is the gastrin, and the secretin and cholecytokinin are the other two hormones which regulate the gastric secretion. Okay, so we see here how these hormones are secreted when the bolus enters into the stomach, and it causes a distension in the stomach wall, and it causes the increase in the gastric juice and also the secretion of gastrin. Gastrin is gastrin stimulates the continuous secretion of pepsin and the hydrochloric acid, and then the acid and also the fat content of the food in the small intestine causes the secretion of the acid and also fat content of the food causes the secretion of cholecytokinin and secretin from the small intestine. And these two hormones has regulatory effect on pancreas and also on the liver. From the pancreas, the bicarbonate ions and also the enzymes are secreted into the small intestine through their ducts. And cholecytokinin is very important on the regulation of gallbladder. The gallbladder is the place where the bile is stored. And when this cholecytokinin stimulates the gallbladder, it releases its bile through its duct into the small intestine, and the bile is going to be used for the digestion of fat molecules. Okay, so how these secretions occur in the stomach and in the small intestine. So there are specific enteroendocrine cells we see here. So this is a small portion from the body part of the stomach, and stomach has three main parts. So the body part is the middle part and the lower part. So this segment belongs to the body part. You see here the cells lined in the this gland. Okay, so these upper cells secrete the mucus and bicarbonate. So mucus and bicarbonate decreases the pH of the stomach, and mucus helps the digestion. So parietal cells secrete the acid, hydrochloric acid, and also intrinsic factor. So intrinsic factor is a very important factor for absorption of B12 vitamin. And enteroendocrine cells, they secrete the histamine, and chief cells, they secrete pepsinogen. Okay, so we see here the hormones which have positive and negative effect on the hydrochloric acid secretion on parietal cells. Gastrin, histamine, and acetylcholine, they have positive regulatory effect. That means they increase the acidity of the lumen of the stomach, and somatostatin hormone, which decreases the secretion of the hydrochloric acid. And pepsinogen is a very important enzyme which is secreted by the chief cells in the stomach. The pepsinogen is required for the digestion of proteins. The chief cells, they secrete the pepsinogen into the lumen, and the parietal cells secrete the hydrochloric acid, and in the presence of hydrochloric acid, pepsinogen is converted to its active form, the pepsin, and then pepsin degrades the proteins into their peptides. And then the digestion of these peptides are completed in the small intestine, and they are converted to their monomers into amino acids. Okay, of course, the gastric motility is very important for the completion of the digestion in the stomach. And the gastric juice, together with the motility, there are smooth muscles in the stomach. So they contract, and they create the peristaltic movement. So this peristaltic movement pushes the food forward and backward, so causing the grinding and the mixing of the food in the stomach, and then converts it into chyme. And then this chyme is then moves to the small intestine for further digestion. Okay, so when we come to the small intestine, the small intestine has a very convoluted and folded structure. So we see here the circular folds, And if we concentrate on this fold, let's say, okay, so this is the villus. So the villi are the finger-like protrusions, and the singular name is the villus. Okay, on this villus, so there are the epithelial cells, and so there are various types of cells covering this villi. So the epithelial cells, which are specialized for the absorption of the nutrients. So these epithelial cells, they have hair-like, very thin protrusions, so they are specialized for the absorption. And also, there are goblet cells. The goblet cells are, they secrete the mucus, lubricating the surface of the small intestine, aiding the digestion. And of course, as we talked, there are enteroendocrine cells which secrete the hormones to regulate the secretion of the hormones. And inside of the villus, you see, there is a capillary network. So this capillary network is required for the drainage of the absorbed small molecules, and then they carry them to the main circulatory system. And in addition to this capillary system, you see here, there is a one-way lymph vessels, which is called as lacteal. Lacteal is very important for drainage of fat molecules, which are absorbed from the epithelial cells of the small intestine. Okay, so as we said at the beginning, the small intestine is connected with the accessory organs of the liver, gallbladder, and pancreas. The liver secretes its bile into the gallbladder, and gallbladder stores this bile. And then when the hormonal stimulation comes to the gallbladder, it releases its bile into the small intestine. And also the pancreas secretes the bicarbonate ions and also the enzymes, which will be used for the digestion of macromolecules such as the carbohydrates, proteins, and the fats, and also the nucleic acids. Okay, so when we look at the secretion parts of the pancreas, we see here, Okay, so this is the duct, end of duct, lobule. So there are the cells which are called as acinar cells. So they are placed at the end of the duct. So they secrete their enzymes into this duct lumen. And also, you see here, there are other duct cells which secrete the bicarbonate. And together, the enzymes and bicarbonate, they are released and travel through this duct and reaches to the small intestine. Okay, the enzymes secreted by this way are, they digest the proteins, the fats, polysaccharides, and the nucleic acids. The enzymes which digest the proteins are trypsin, chymotrypsin, and carboxypeptidase. So these enzymes are secreted in their inactive forms. They are activated in small intestine for digestion of these macromolecules. Okay. You see here how the trypsin is activated. The enzyme, the trypsinogen, is secreted by the pancreas. And then when in its, you see the trypsinogen is the inactive form of trypsin. And then when it reaches to the small intestine, so there are the brush border enzymes. So these brush border enzymes, they are called as enterokinase enzymes. They activate the trypsinogen into trypsin, and trypsin activates other inactive enzymes, including the trypsinogen and also other inactive enzymes like chymotrypsin and also carboxypeptidase enzymes. Okay, so liver, we said it is very important for secretion of bile. So why bile is important? Because bile contains the bicarbonate ions, which decreases the acidity in the small intestine because the chyme contains very high acidity, and also cholesterol, phospholipids, bile pigments, organic wastes, and also bile salts. So these bile salts specifically, and also the other the components of the bile, so they are very important for solubilization of the lipids. And the bicarbonate ion neutralizes the acidity. Okay, so this bile in a day, during the digestion of a meal, several times the bile is secreted from the liver and it is returned back to the liver. Okay, so let's say the bile is secreted into the gallbladder, stored there, and then secreted into the small intestine, and then it is used for the digestion of the fat and the ileum part of the small intestine. So this is the part where the bile is reabsorbed. Okay, it is absorbed and it enters to the hepatic portal vein. So this portal vein is the vein which carry the bile back to the liver. So the recycling of bile between liver and the small intestine is called as the hepatic circulation of bile salts. And very small amount of bile is lost, about 5% is lost with the feces. Okay, so how the macromolecules are digested in the small intestine. Let's begin with the carbohydrates. You know, the carbohydrates are made of they are the polysaccharides. Their digestion begins in the mouth with the salivary amylase. The salivary amylase break down them into small fragments. And then when it reaches to the stomach, due to acidity, this amylase enzyme becomes inactive. And in the small intestine, there are the enzymes which are called as pancreatic amylase. It continues the further digestion of these fragments into smaller parts. And then at the brush border of small intestine, there are enzymes, specific enzymes that break down these polysaccharides, the disaccharides into monosaccharides, such as sucrose, maltose, and lactose are degraded by their specific enzymes into their monomers. And then they are transported through their specific transporters into the epithelial cells. And at the other side of the epithelial cells, they enter to the bloodstream, and then they go to the liver. And in the liver, so mainly they are converted to glucose in case of these monosaccharides, or they are stored in the liver as glycogen, or they become part of the blood glucose, or they are stored as muscle glycogen, or they are converted to fat and used later on. And how about the digestion of proteins? Okay, the digestion of the proteins begins in the stomach by the pepsin. The pepsinogen is converted into pepsin, as we learned in the presence of the hydrochloric acid. Okay, the pepsinogen is converted to its active form in the presence of hydrochloric acid, and then it breaks down Okay, the hydrochloric acid denatures the proteins, and then pepsin breaks them into small fragments, and in the small intestine the pepsin becomes inactive due to high pH, and the other enzymes secreted by the pancreas, and these are inactive, so they are activated by enteropeptidase enzyme. And enteropeptidase enzyme converts the trypsinogen into trypsin, and trypsin then activate other inactive enzymes. And then trypsin and chymotrypsin breaks the internal bonds, and carboxypeptidase breaks the end amino acid, the bond between the last amino acid and the previous one. And at the end, we have free amino acids, dipeptides, and the small fragments. So the further digestion is carried out by the microvilli brush border enzymes. The dipeptidases, which break down the peptide bound between the dipeptides, and aminopeptidases break down the bond at the amino terminus, and then they are transported through their specific transporters into the intestinal epithelial cells. And at the other side, they are transported into the capillaries like the monosaccharides, and then they go to the liver, and then they are taken up by the liver cells, or they are used by other body cells, or they are converted to glucose, or used by the cellular respiration. Okay, so the last macromolecules are the fats. So how the fats are digested? Okay, lingual lipase is secreted by the saliva in the mouth, but it is inactive in the mouth. So these are the triglycerides. And in the stomach, the other gastric lipase is secreted. The gastric juice partially digest the fats, but and the 30% is digested. But since they are water-fearing molecules, they form very large lipid masses. So this should be solubilized into small small particles. So the bile salt has the hydrophilic, and also which is released by the gallbladder, which has hydrophilic and also the hydrophobic property. So this hydrophilic property attracts the fat molecules into the water, and hydrophobic parts, they enter into the mass of the fat and separating them into small droplets. And the pancreatic lipase, it carries out the further digestion and separating them into their fatty acids and monoglycerides. And then, since they have hydrophobic property, they can easily diffuse through the small intestine. And in the small intestine, they are resynthesized into triglycerides. And in the endoplasmic reticulum, they are converted to lipoprotein called as chylomicron. The chylomicron has a very huge size. So because of their huge size, they cannot be taken by the capillaries, and they are taken through the lymph vessels. So through the lymph vessels, eventually they enter to the bloodstream through the vein. And in the circulatory system, they are used for further usages. Okay, so the last part, digestion in the small intestine, is the digestion of the absorption of the vitamins. Vitamins are small molecules. They do not require any digestion. And depending on their structures, the fat-soluble vitamins such as A, D, E, K vitamins, they follow the same pathway for the fat absorption like other lipids. And the water-soluble vitamins, they are absorbed by the small intestine by diffusion or mediated transport, except vitamin B12. Remember, we have learned that in the stomach, the intrinsic factor It's a protein which is used as a transporter of vitamin B12, and it is taken into the epithelial cells of the small intestine by endocytosis. And water and minerals, of course, they are absorbed either by diffusion or by channel proteins, and the other ions were crucial for our physiological functions of our cells, such as the sodium ions, chlorine ions, potassium and calcium and irons are absorbed by the small intestine. And after absorption, they follow different pathways. The fat molecules, they enter the lymphatic system, and later on they enter into the circulatory system. But the water soluble molecules, which are absorbed in the small intestine, in large intestine, and also some molecules such as the hormones from the pancreas or from the stomach. So they are carried to the liver by hepatic portal system. And during the digestion, of course, the motility of small intestine helps the digestion of the food. There are two types of motility occur in the small intestine. The peristalsis, so which pushes the food forward, and the segmentation, the circular contractions of the smooth muscles on the small intestine causes both the mixing and also further pushing of food to the large intestine. And okay, so this is the large segment of the gastrointestinal system. The large intestine has little or no digestive function. So it is mainly absorbed water and electrolytes such as the sodium and chlorine ions, and also secretes mucus containing bicarbonate and potassium ions. But it is very important in the synthesis of some vitamins such as vitamin K, because the large intestine has a very special flora, microorganisms, so they synthesize the vitamins for us. And also recently, so it has been discovered that the large intestine has very important function to increase our immunity. And the forms the feces and carries out the defecation. Okay, thank you very much. Okay, this is all for today. Take care. Bye-bye.